Okay, hi everyone. Happy Friday. Let's continue on with where we left off last time when we were talking about how B cells, when they enter a lymph node, and these naive B cells that are mature and are, working by going through our blood and our lymph, but naive, meaning that, they're looking for something to bind, but they haven't bound anything yet. We talked about how when they enter something like a lymph node, they are presented different antigens from these large follicular dendritic cells, and then these B cells, if they actually see something that they can bind to with their immunoglobulins, right, then these B cells will seek out helper T cells that have also similarly entered the lymph node, been presented different types of peptide signals by things like common everyday dendritic cells, and now these helper T cells and B cells will pair up, right? And then this pair of lymphocytes will then proceed to kind of migrate around inside the lymph node, either towards the medullary area where the lymph fluid exits. And so some of those T cells will tell those B cells to become plasma cells. They can start making antibodies, but at the same time, some of the T cell B cell pairs migrate up to the primary follicle region of the lymph node, where they then undergo hypermutation. So let's continue examining this. So this is the scenario that we've been discussing. Right? So the lymphocytes, a B cell, T cell, that enters a lymph node either through the blood or through the afferent lymphatic vessels, because maybe they were already in the lymph system at some other lymph node, and now they're flowing into this one. Up until this point, I was kind of joking last time that, like, whoa, this B cell, this T cell, they don't realize that they're actually sort of meant for each other, right? Because they both have the same antigen recognizing either immunoglobulin on the B cell or T cell receptor on the T cell. But until they've had a chance to bind those signals, they don't care about each other, right? They're just like kind of floating through your system together. But now that they've arrived at this lymph node and both of them are presented with signals that they recognize, then they pair up. And then they do this kind of little dance that I was talking about, where the helper T cell will encourage the B cell to make many, many copies of itself. Some of them will then be encouraged to become plasma cells that will then drain out of the lymph node in the lymph fluid, producing big, big clouds of the free-floating antibody form of immunoglobulin. In this case, it would be, let's say it's the first antibody that's made from this particular B cell clone. So it would be an IgM, right? A free floating antibody version of the IgM. And that goes off, and that's your first sort of quick and dirty antibody response, right? Because up until this point, you haven't had any antibodies that could recognize the particular pathogen that is causing this infection to happen. Meanwhile, we said that some of these B cell clones are encouraged to go to the primary follicle areas, where they start creating these germinal centers, centers where they're now dividing and undergoing somatic hypermutation to try to randomly tweak the light chain genes that they have to maybe improve the fit of their immunoglobulins, right? And so that's where we left off last time. So let's take a look at these germinal centers and see what they kind of look like. Right. So here you have a really neat image. What researchers did here was that they realized that in a given lymph node, so this whole big structure here is a lymph node, right? You would have multiple germinal centers, or I should say multiple regions of primary follicles where you could have germinal centers starting up by B cells that are rapidly dividing and mutating. But if you consider that in the face of this infection, if this lymph node has different antigens that are coming from the source of infection, it's not like there would be just one specific antigen, right? There'd be all kinds of molecules coming from something like the virus or the bacteria that would all be representative of things that your body normally doesn't have. So each one of these different types of molecules coming from the pathogen would represent a potentially good target as an antigen to be selecting B cells, right? Because at any given time, you've got like, hundreds upon hundreds of millions of B cells coming out of your bone marrow, and they all have different sort of IGs. It's not like you're really looking for just one particular B cell clone. You're looking for a bunch of different B cell clones that all have IGs that are useful, right? That all sort of have close enough fit to one or more of these different antigens, or maybe the same antigen, but different epitope three-dimensional structures on that antigen that can bind different immunoglobulins. So if you think about that, at any given time during an infection, you would have many different B cells that have been selected, and many of these B cells then would be up in these germinal centers together, all busy doing their hypermutation, even though their immunoglobulins are targeting different things, right? But that's okay, because the dendritic cells, like the follicular dendritic cells that hang out in those germinal centers and are constantly showing antigen to these B cells, right? They're showing a wide array of different antigens, right? Not just one. So even though we simplify things in many of our illustrations, where we tend to conveniently say, oh, look, there's a red dot, right? And that red dot represents the antigen. The reality is a dendritic cell that's presenting antigens would be presenting many different kinds of antigens, and therefore would be communicating with many different B cell clones. So what these researchers did was they came up with a series of different antigen molecules that each sort of were labeled with a different fluorescing dye, right? So therefore, wherever you would see, these antigens being sort of concentrated or focused on, then you would see these different colors. And they went ahead and during the course of an infection, they went ahead and looked at these germinal centers. And what do you see? So each of these dots here basically represent particular B cell clones that are in these germinal centers undergoing hypermutation. Right? So at first, you might imagine that a germinal center would have many different B cell clones that were all selected against different antigens. That's why you see a sort of smattering or wide range of different colors. Right? These all represent the antigens that are being focused on by the different B cell clones that are hanging out in this particular germinal center. But as hypermutation progresses, you can easily kind of imagine that certain B cells against certain antigens might be more successful in hypermutating and getting better and better and better. And that means the population of B cells in the germinal center that you're looking at would start become imbalanced, right? Instead of being a mix of all these different B cell clones, right? Over time, you would see those particular B cell clones that are being really successful and having good antibodies against their antigen. They're the ones who kind of take over, and they're the ones who become like the focal point of all the Ig production. So over time, what do you see? Like in this germinal center up here, it's turning more and more green, right? Meaning that the B cells that were up in this germinal center during their hypermutation, the ones that bound to, let's say, the green fluorescing antigen, they're the ones who are really taking off, right? So it's almost like this germinal center is a little sub area where B cells that are getting particularly good at immunoglobulins that recognize the green antigen, that's where they're getting better and better, right? Over here, maybe on the left, this germinal center is turning more and more red, right? Meaning you would have certain B cell clones in this germinal center that are getting really, really good at detecting the red antigen. So therefore, they're the ones that are encouraged to keep hypermutating. So this particular germinal center is like a little sub area where the antibodies or the B cells that know how to make the antibodies against that antigen are coming out of, right? So think about it that way. And then that way you appreciate that in the face of an infection, right, your body is actually selecting for many B cell clones as starting points because each of them just by chance had close enough fit IgMs on their surface, right? And now over time, you're encouraging those to kind of hyper evolve so that you can make really good quality antibodies against different antigens. If we take a closer look at the activity that's taking place inside of a germinal center, this is what we see. So the B cells that had paired up with helper T cells, because both the B cell and T cell recognize the same antigen signals, although remember, it might be different uh, molecules that the immunoglobulins on the B cell recognize versus the peptides that the T cells recognize. But we said last time that if you consider that bits and pieces of antigen are probably complex little chunks of things, of molecules that have some protein, some carbohydrate, maybe some lipid, because they came from a fragment of the bacteria, right? We said that even if the Ig of a B cell might be picking up the carbohydrate part of this chunk of material, once it brings it inside the B cell and it processes it, right, whatever little protein bits of that chunk of the bacteria are converted into small peptides, that's what gets presented potentially for a T cell to see, right? So that was that concept of linked antigen recognition between B cell and T cell. So remember, some of those then paired up B cell and T cell will be going up into the primary follicle so that the helper T cell is essentially telling that B cell, hey, I'm going to kind of accompany you up here and drop you off because you should start doing this hypermutation. So those B cells become a very specialized type of B cell known as a central blast. So they're pretty big, right? And importantly, at this point, the central blast stops making immunoglobulin on its surface. Why? Because these central blasts are going to participate in hypermutation. So it's almost like they put a pause on making the Ig that their cookie recipe that they've been carrying around codes for, right? They stop making the Ig product so they can focus on hypermutating the Ig genes. So as they hypermutate, they then divide and form daughter cells. And these daughter cells begin to re-express the Ig on their surface, right? Presumably, this is now the slightly tweaked version of the Ig through random mutations that were introduced at the Ig light chain locus. So these centrocytes then, right, begin to coat themselves with that Ig, and these centrocytes then test out their new version of the Ig against the antigens that the follicular dendritic cells, who are sort of hanging out here with them. So if we look at the cartoon here, right, these big orange stars are the big follicular dendritic cells that are constantly sort of showing the antigen, right? It's like they're always there to constantly remind these dividing B cells, hey, remember, this is the antigen that sort of like brought you here in the first place, right? This is it. You have another chance to look at it. How does your new Ig compare to your old Ig? So if the new Ig that these centrocytes have on their surface, right, actually ends up binding better or stronger to the antigens that are being presented by these FDCs, the follicular dendritic cells, well, that means these centrocyte B cells are spending more time attached to the FDC cell, the follicular dendritic cell. Therefore, they are receiving stronger cytokine survival signals compared to maybe another central blast, big one, doing hypermutation. It tweaks its Ig, so it divides into two centrocytes. The centrocytes start expressing the new version, the tweaked version of the Ig on their surface. Oh, but guess what? Because of the random mutation method of trying to tweak them, the shape of the Ig is actually worse than the original B cell that came in there, right? And that means they're not going to bind to the antigen as well that's being presented by the follicular dendritic cell. They don't receive nearly as much of these survival signals from the dendritic cell as a result, right? And then these B cells, you don't need them anymore because they ended up being worse than what you started with. So as a result, higher affinity B cells take over the germinal center. That's why in the previous slide, even though the germinal center might start off being multicolored, the different colored dyes representing different B cells that are all in there trying to do hypermutation, over time, right, certain B cells become better and better, and therefore they take over the germinal center, and that germinal center becomes this little sub area that kind of like specializes in B cells that just focus on that one type of immunoglobulin or antibody, right? What was I going to say? Meanwhile, what are those helper T cells doing? The helper T cells that kind of accompanied those B cells up there in the first place. They're hanging around also, right? They're hanging around on the perimeter here because as centrocytes are surviving, right, and moving out because they have better immunoglobulin than what they started with, those T cells are there to meet them, right? And they pair up, and then some of those T cells then take those better centrocytes back out into the lymph node where the process repeats, right? They pair up, and that B cell is told to make many copies of itself. Some of them then turn into plasma cells. Right? Some of them are sort of escorted back up into the germinal center so that this process keeps going and going. Right? After the infection is done, then you don't have a need for a germinal center like this anymore. The germinal center will shrink, and you're left with B cells that represent the best B cells that you came up with during this bout of illness, right? And those are the ones then that will then travel out of the lymph node and go to places like your bone marrow, where they will persist. They will hang out as your memory B cells, right? That carry with them as their starting point of immunoglobulin, the best version of immunoglobulin that you came up with, right? As a result of this prior infection. All right, so let's see. This is another way of sort of representing what's going on. You have helper follicular T cells. So remember CD4 helper T cell of the TFH subtype, the follicular helper T cell. It pairs up with a B cell in that T cell area, the boundary between the cortex and the medulla of the lymph node, right? That's where they're pairing up and where they do their little dance. And this B cell, right, that bound to antigen that was being presented by something like the follicular dendritic cell, it processes the antigen and it presents the peptide part that the helper T cell likes, and that's why they have found each other, right? So then this pairing then might migrate up into the germinal center, where the follicular helper cell says, okay, I sort of escorted you up here, right? And hypermutation takes place. And what happens now? So this panel here represents a centrocyte that has already undergone that process of hypermutation. So the B cell that was sort of moved up there became a big central blast. It stopped making Ig so it could focus on hypermutating, and then it divided into two daughter cells, the centrocytes, that began to re-express immunoglobulin on their surface, right? So let's look at this scenario here, right? Oh, no, sorry. Let's look at this scenario here, right? If the tweaked Ig that this daughter cell has, oh boy, right? The random mutations made it such that it actually doesn't bind the antigen anymore. Oops, right? So not only then will it not really hang out and receive survival signals from the follicular dendritic cell, but look, as a result, the T helper cell, right, has lost interest in the B cell as well, right? So they had met up, there was like this wonderful kind of rendezvous, but now ultimately the resulting B cell here, right, is no longer of interest to that T cell. So unfortunately, without those survival signals, that B cell is just eliminated, right, undergoes apoptosis, right? It's kind of sad. It's like this whole teenage drama thing. But if, okay, happy story here, right? If the centrocyte, its Ig, the tweaked version through hypermutation, actually binds to the antigen stronger, right? That means it associates with the follicular dendritic cell that's presenting the antigen more. It receives a lot of survival signals, and look, right? The follicular helper T cell, right, sees that this B cell is doing a really good job now of binding and presenting lots of the little red dot, because its immunoglobulins have gotten better and better at being able to pull the antigen into the B cell. So therefore, this pair then goes back into the lymph node, and the T cell tells the B cell, go ahead and make many copies of yourself. And then some of those become plasma cells, right, that then start releasing the better version of the Ig. Meanwhile, some of these are escorted by the T cells back up into the germinal center, where they begin or they continue the process of hypermutating to try to see if they can get the Ig even better than they did at this point. So thus, during the course of your illness, we said that the very first wave of free-flowing antibodies that are coming out of your lymph nodes were just simply that first kind of iteration or first version of the IgM that your B cell had when it was selected, right? That's the close enough puzzle piece fit. It's kind of a wimpy fit, but it's better than nothing. So you might as well start sending this out into your system as antibodies. But due to all of these rounds of hypermutation and selection, you are getting wave after wave of plasma cells being sent out of your lymph nodes, each wave producing better and better immunoglobulins. Up until the point when you reach immunoglobulins that are so good, right, then they're really effective at targeting the pathogen, and that's what saves you, right? Because that's what ultimately then is your adaptive immune system saving your life. And then once the infection is over, then back in the lymph node, the germinal center shrinks away because there's no more hypermutation needed. And you're left with those B cells that have the best immunoglobulin cookie recipe that you came up with after tweaking it over and over and over again. And those are the ones that stick around as your memory B cells. So that means when there's an active infection going on and your immune system is like waging war against the pathogen, right, versus after the infection's over, there must be some difference then in what the T cells are communicating with the B cells. So on the left is a scenario where you're still in the middle of actively fighting an infection, right? This is not when you're going to let down your effort. You're trying to ramp up as much high quality immunoglobulin as possible. So during this situation, when the CD4 helper cell, the follicular helper cell, is paired up with the B cell, it is sending IL-10 cytokine signals to the B cell. Remember, quick review. This T cell recognizes the peptide that represents the protein component of whatever this big chunk of antigen is that this B cell recognizes. I mean, could it be that the B cell's Ig recognizes the same peptide as the T cell does? Yeah, it could be, right? There's nothing wrong with that. In fact, that's kind of like almost an ideal situation. But more realistically, right, this red dot piece of peptide is just whatever protein component is part of this big chunk of thing here. And maybe it's lipid or carbohydrate that the Ig actually sees, right? In any event, because they pair up, the helper T cell expresses CD40 ligand so that the CD40 receptor on the B cell can pay attention and realize that the T cell is saying, hey, I'm about to send out some messages here, and these are meant for you and you alone, right? And so these IL-10 messages then are received by this B cell, causing this B cell to differentiate into, for example, plasma cells, right? To start releasing that Ig as an antibody. Okay, so during the active battle against the infection, the priority is to select for B cells to become plasma cells so you can make antibodies that fight and terminate the current infection, right? I mean, hey, it's no use or there's no point worrying about memory here if this thing's going to kill you, right? You need to survive and beat this thing first and then worry about remembering it. So that's all a function, right, of the cytokine soup inside the lymph node that this T cell B cell pairing is taking place in. Right? Because it's that cytokine soup that actually tells the helper T cell, hey, we are right in the middle of the infection right now. We need to get as many B cells differentiated into plasma cells as possible. And that's what makes the T cell send out IL-10 to the B cell. However, on the right here, as you are successfully beating the infection, and as a result, there's lower and lower and lower evidence of infection going on inside of your body. That means you have lower and lower and lower cytokines being released by infected tissue somewhere else in your body. So the soup of cytokines inside the lymph node, where the T cell and B cell are pairing up, right, reflects that, reflects the fact that, yeah, you know what, there's no real active infection anymore. It's almost done. So that environment encourages the T cell to send these little IL-4 blue triangles as the message to the B cell. So it says, no, we don't need you to turn into a plasma cell anymore because I think we're all good, right? We're all done. You should start turning into memory cells, right? So these memory cells then represent B cells that are not differentiating any further right now. They're just sticking around, right? So when everything's done, you still have these memory B cells, importantly, with the best tweaked Ig recipe, right, that you got from going through these multiple rounds of affinity maturation through hypermutation. And so this then represents the investment that prevents future infections from causing disease. Right? You're going to have a pool of these memory B cells and some memory plasma cells, right, that also stick around, so that when that same pathogen comes back again, right, this is where you start with now, not square one, right, but you start off with really good high affinity IgGs. All right. So part of understanding how B cells respond, or what a B cell needs to do in order to release the right type of antibody to fight infection, has to do with understanding some of the differences between these different immunoglobulin molecules that a B cell can make. And understanding also that the types of cytokine messages that a B cell will receive from the T cell that's communicating with it, right, will actually drive what type of immunoglobulin the B cell switches its recipe over to making. So we said that if you look at this table here, so here are the 5 types of immunoglobulin subclasses. There's IgM that all naive B cells start off with, right? Then you have IgA, IgE. They don't list IgD on here because the IgD actually doesn't really function as a pathogen fighting immunoglobulin. The IgD functions more like a kind of regulatory Ig when the B cell first emerged from the bone marrow and it was undergoing tolerance training in the blood. We discussed this briefly, that at that time, it's making both IgM and IgD on its surface. That's the one time it can make 2 different Igs, right? 2 different types. And that IgD plays a role in making that B cell go anergic or shut down if it fails tolerance training. So for the sake of this table here, this table is actually talking about switching to different Ig types during an infection and what cytokine messages from something like a helper T cell trigger that. So in the previous slide, we said, for example, a paired T cell with this B cell might talk to the B cell using IL-4. Right? And so IL-4 then, what it would do is it would inhibit further production of IgM and encourage the B cell to switch to making a different type of Ig, for example, IgG1. So you notice there are 4 subtypes of IgG here, right? So not only are there 5 isotypes of immunoglobulin, M, G, D, A, and E, yeah, 5, but within the IgG type, there are subtle different versions. So IgG1, 2, 3, and also an IgG4. Right? All with subtle different structural features that make them better or worse for certain applications. IgG1 is the one that we usually think of when we say IgG, right? Because this is the one that B cells tend to isotype switch over to early in an infection. Right? So it's the local lymph node cytokine environment that instructs the helper T cell what's going on at the site of infection. Either there's a raging infection going on or there's not much of an infection anymore. And importantly, the type of infection, viral, bacterial, parasitic, etc., so that the helper T cell knows, how to help other immune cells like B cells become what they need to be. Right? So therefore reflecting the specific need. And then that will determine what cytokine messages the helper T cell sends over to the B cell. So remember, IgM is considered the first quick antibody response, but very quickly, as you're starting to do more and more B cell selection and hypermutation, the IgM production is shut down, right? Because you don't want to, once you've come up with better versions of the Ig, why do you want to waste your time still making the sort of crummy, close enough fit kind, right? You want to focus on making better and better ones. When switching from IgM to IgG or A or E, right, B cells can only make one Ig at a time. Right? And once they become fully differentiated as a plasma cell and start pumping out tons and tons and tons of this particular type of Ig isotype, like A or E or G, as we said before, right? That plasma cell, that beast of a cell that just becomes a huge factory making proteins, it can no longer switch, right? Because it's kind of done, right? But so it finishes its life as that plasma cell making that particular IgA, G, or E, right? And it gets replaced by another plasma cell that presumably has a better version of that IgA, E, or G to send out. Oops. Okay. So this cartoon, I think, is really useful to help you review. Because, one thing I try to encourage you in this class is to try to put things together, right? I think we all know the better you can put things together and actually make in your mind almost like a story, right? Of how story has a plot and certain things happen, there's characters, certain events happen. The more you can kind of create the story in your mind, the way better chance you'll have in actually remembering all the details of what cells are involved, what events are involved, and little molecules are involved, like these cytokines, right? So I think this is the type of figure that helps you do that. Let's look on the left. Here we have an infection taking place, all right? But maybe there's two different types of infections at different times. So in one scenario, you have a viral infection. So these green squiggly things are virus particles. And in another scenario, so this might be taking place, for example, in your skin, like a breach in your skin, for example, that lets virus in. In a second scenario, you have a multicellular parasitic worm, right? So this is not just a teeny tiny virus, right? This is like a worm that's made up of 1,000 cells or more, right? Each cell that makes up the worm is already like 1,000 times the size of an individual virus, right? So very different scales here, right? So that happens maybe because you ate some undercooked food or something like that. And so in your intestines, so this is the lining of your intestines, you have this parasite coming in. So these are definitely two different pathogens that both need to be dealt with by your adaptive immune system. But obviously, the response of your immune system is going to be very different. Right? In both cases, bits and pieces of antigen, so evidence of the pathogen, are draining into lymph nodes, right? Draining to lymph nodes. And so they arrive at, let's say, the closest lymph node to where this is happening. And this is where then T cells and B cells will be activating. So the T cells that get to activate against this antigen that's coming in, there is a whole soup of different cytokines in this lymph node that reflect, right, the types of cytokines that are released by virally infected cells under your skin that are yelling for help, crying for help, versus the infected cells that are being damaged by something like a big parasitic worm, and they're crying for help, right? Different cytokine messages that create the soup or the environment inside the lymph node that reflect the nature of the infection and therefore inform these T cells as they're activating, what the threat is and how they should begin to mount a defense. In the case of CD8 positive T cells. I already said CD8 cytotoxic T cells, they sort of have like a one-track sort of mind, right? Once they activate, right, they just go off and are basically like, okay, I'm going to go bump into other cells now. And if those other cells that I bump into present the peptide signal that my TCR recognizes, I'm just going to go ahead and tell that cell to die, right? So that's what you see here. The cytotoxic T cell has synapsed with an infected cell that's presenting the antigen on MHC1, and therefore the cytotoxic T cell sends out all of its sort of very nasty cytotoxic cytokine messages that tell this infected cell to undergo apoptosis and die, right? As a way to stop or slow down the spread of something like a viral infection, right? On the other hand, you will have a naive CD4 helper T cells that have also wandered in here, and they're activating as well, right? And so when the activated helper cell is activated, it needs to decide, right, which subtype or class of CD4 helper cell should I be? And remember, this is all a result of the soup of cytokines in the lymph node telling the activated helper T cell, is this a virus or is this a parasite that we're dealing with? And that will then encourage that cell to turn into either Th1 or Th17, Th2. Some of them will be encouraged to turn into follicular helper cells so they can hang out in the lymph node and start talking to B cells. Others will be converted into Treg cells so they can go off and try to keep the response sort of under control, right? Make sure that the immune response doesn't get out of control, right? So ultimately, these helper T cells, like the follicular helper T cells, stick around, will be pairing up with B cells that also wandered into this lymph node and are activating, right? And then depending upon the nature of the infection, the helper T cell will tell this B cell, hey, first, some of you need to turn into plasma cells so that you can make the IgM form of your immunoglobulin, right? Because your immunoglobulin works to bind this thing that we are detecting, right? That's coming from the threat. Also, other signals like IL-4 might tell some of these B cells, you go ahead and class switch, right? We need to have things like IgG made or IgE made. Now, this cartoon's a little inaccurate, right? Because one single plasma B cell would not make IgE and IgG, right? I think all it's saying is that in this case, it's making IgE, but other plasma cells are releasing IgG as a result of class switching. Or it may say the nature of the infection is such that you should make IgA. So, for example, you see here, IL-5 messages coming from the helper T cell encourage B cells to become fully differentiated plasma cells. That's that class switch from IgM over to IgA, for example. All right, so this brings us to our discussion about, what is it about an IgA, an E, or an A, or a G that makes them more useful for certain types of situations? This is just a reminder of the 5 subtypes or classes or isotypes of Ig. Remember, a B cell starts off making IgM with its unique cookie recipe that gives it a unique antigen binding domain to its Ig, so the red part. If it class switches, right, if a T cell tells it to class switch, it will keep the red part the same, because after all, that's the part that matters, right? That's why this B cell is special, because it knows how to make this particular red part. But the stem part will change, and these are those constant blocks that are added on to make the complete Ig chain genes. Since immunoglobulins are little proteins, remember they're like 150 kilodalton proteins, and not whole cells, then you can actually make a massive amount of these free-floating antibodies at any given time. Right? And these can then go flowing through your blood, and they have a powerful target-specific, like swarming type of activity. Yeah, it really is like this poor guy over here who is attracting the attention or the ire of the swarm of bees, right? Even though each individual little bee is tiny compared to this poor guy here, if you have a whole swarm of them, it can be an overwhelming response. So these Igs are also very useful. Antibodies are also useful because they can directly destroy or interfere with the pathogen, in which case, if it's these Igs alone that are working without any other cells to destroy the pathogen, and the Igs alone or working with things like complement proteins, right? Because Igs will actually be able to attract complement proteins. Then we just call this humoral immunity, right? It is cell-free immune response, an immune response that is driven purely by these proteins. Other antibodies can function by recruiting other cells to bring the attention of these other immune cells to be directed at the target. So these are all advantages of antibodies, right? Immunoglobulins and antibodies. And you can see why then these represent sort of the latest, greatest invention of our adaptive immune system in terms of evolutionary history, right? Because remember, B cells are sort of the most recently evolved cells of our immune system, right? So therefore, their ability to make antibodies, these little antibodies, are really this great little invention that has enabled us now to have much stronger responses and, importantly, memory. So IgM is the first antibody that's released by the activated B cells. I think I showed this to you before, or something similar, right? The IgM antibodies actually associate, so 5 of them come together to form a ring, and they're sort of held together by this little, what we call a J protein. And the IgM is first released in response, right, to the first sort of detection of pathogen and selection of B cells. Each one of these antigen binding domains is pretty wimpy, right? Because this is the random puzzle piece analogy here. But if you have 10 of them, because you have 5 Igs in a circle, then they become very strong, right? You can actually have strong binding, and they will bind to then the surfaces of a pathogen, and then recruit complement proteins to opsonize the pathogen, right? And you might think, well, that's kind of weird because I thought complement proteins should bind the pathogen itself. Yeah, I mean, that's why you have complement proteins. That's why your innate immune system relies upon the fact that these complement proteins, like complement protein C3, is usually good enough to detect and opsonize something like a bacteria on a thorn that got stuck into your skin. But the whole point is, what if you have bacteria or virus particles that complement protein for whatever reason isn't good enough to stick to? That's how you even got to this point in the story where your B cells needed to be selected for so that you could make antibodies that would then bind to the bacteria, and then the complement proteins bind really well to the antibody, right? So the antibody is helping the opsonization of complement proteins happen. So the advantages are, it's very quick to make and release these IgMs. The other advantage is, even though each antigen binding site is kind of wimpy on it, I say why? Well, we already discussed that, because these are just the B cells that were selected to start off with based on close enough fit. Overall, because you have 10 sites on there, the binding is pretty strong, right? And it's also an advantage because this is the primary way that the classical complement pathway, right? Remember, there's 3 pathways of complement activation: alternative, lectin, and classical, right? So the classical, when we say it involves antibodies, this is what we're talking about. Right? But the disadvantages of IgM is it's really bulky, right? Because you've got this big ring. So it's not very easy for this complex molecule to exit the blood to enter different tissues, right? So primarily, you have a lot of IgMs in your blood, or doing their thing in your blood, right? So IgGs and IgAs provide higher binding affinity and flexibility, right? Because after B cells have started undergoing hypermutation and you have better versions of the Ig recipe, then those B cells that have that updated recipe, if they're turned into plasma cells, they may also be told, all right, forget about making the IgM version here. Just go ahead and class switch and make the IgG version using this new improved antigen binding domain, right? So IgGs are flexible. They're the best sort of all around antibody to have, because they are compact and they have this flexible hinge region. So these little black lines, I think I mentioned before, represent disulfide bonds between cysteine residues on the left and the right heavy chain. Right? And so that holds the Y shape together. So that hinge region is flexible so that the two arms of the Ig can actually move around pretty freely. So you can bind like two different antigen or two different instances of the antigen on the same target, or you could have two different targets that each have the antigen on it. And then this IgG could be involved in doing something like getting these different antigens to stick together. Right? Those of you who are taking the lab right now, next week, you'll do an exercise where you actually use IgGs to cause things like red blood cells to come together in solution. So you can really appreciate how these IgGs can cross-link different surfaces. Dimeric IgA is also advantageous because it is smaller than the big IgM ring. And in this case, two IgAs come together, held together by the J chain protein. Right? Oops, sorry. And I should say that most IgG, or most of the Ig that's in your blood, is actually the IgG type. Right? So IgG, just like any other antibody, is a protein, right? And most proteins don't just float through cell membranes. Proteins have to be somehow transported across cell membranes, right? Because proteins, they're just not going to like meander their way through the phospholipid bilayer. They have to be actually transported. So the transport of things like IgG across a membrane is facilitated by special proteins that function as receptors on the surface of the cell. So in this case, there are a group of proteins known as Fc receptors. What does Fc mean? Remember, the stem part of an immunoglobulin, right, is also called the Fc region. Right? So Fc receptor, not surprisingly, means that this is a receptor protein here that recognizes and binds to the stem region of a particular isotype of Ig. So in this case here, this FcRn, if there's IgG floating by in the blood, so that's why they bother having a red blood cell here to remind you that this white space is the blood, and this is the endothelial cell lining your blood, right? They can actually bind to this and pull them through and deliver them to the other side of the membrane. Okay. So IgA is most useful in the linings of our GI tract, our eyes, our nose, throat, basically places where you have a lot of mucosal tissue, where exposure to pathogen might come from being breathed in, for example, or swallowed. IgE is the best form of Ig to fight against large pathogens. Because again, the IgE is a very sturdy form. There's no hinge region at all, so it's not easy to break apart. So this provides a very sturdy structure to bind to things like large multicellular parasites, like these different types of roundworms, hookworms, tapeworms, nematodes. And IgE, importantly, does not function by being a free-floating antibody. Right? It functions because certain immune cells, like mast cells or basophils or eosinophils, have certain receptors, Fc epsilon receptors on their surface that can hold onto the stems of these IgEs. Right? And when that happens, it stimulates the particular immune cell, the mast cell, to release its granular contents. Remember, these are all granulocytes, so they have the granules in them, and these granules are filled with compounds that are oftentimes very irritating, right? Things like histamines that cause your tissue to react kind of violently. Right? And this way, it causes your tissue to respond in a way to try to flush out the parasite. So whether you're talking about sneezing, or coughing, vomiting, or even excess mucus production, right? These are all things to try to attempt to physically get the offender out. So I know I'm running out of time. So last couple slides real quick. Right? So here you have an inactive mast cell that's filled with these granules. Right? On its surface, it has the green Fc epsilon receptor that hangs onto IgE antibodies. Right? As a result, when things bind to the IgE, so this red thing here is an antigen, and in the case of these IgEs, it requires a couple of these to actually co-bind to the same antigen that will then stimulate the mast cell to release its granules. So here you can see an activated mast cell due to binding antigen has now released all of its granules, right? And all this like histamine and other noxious chemicals are coming out. Eosinophils are a good example of these granulocytes also. So here you have a larva of some kind of a parasitic worm, so a schistosome, right? And on its surface has these red triangles, the particular antigen that the IgE recognize. And you can see the IgE are being held onto by these eosinophils through its Fc epsilon receptor. And what that allows is that these eosinophils that normally would be sort of innate effectors, they don't have any targeting ability. If they're stimulated, they just blow up and release these noxious contents, because now they actually stick to the surface of this parasite because they're hanging on to IgEs. Look down here, right? This is cool. This is the actual larval schistosome here. All of these are eosinophils that are stuck to it now because the eosinophils are hanging onto IgEs that recognize antigen on the surface of the parasite, right? So these innate effector cells, like these eosinophils, right, that normally don't know like what a target is, they're just stimulated and they go ahead and release a big cloud of this stuff. They suddenly take on more powerful targeting abilities. They become adaptive responders because they're able to utilize and be suited up with IgEs, right? So it's kind of like this guy here. Right? This gives them the power now that's a lot more powerful than if they were just left alone as being just eosinophils floating around. Right? Okay, let's go ahead and stop there. Sorry, I went a little over.